Here we will continue our discussion about this differential expressed genes in the aged CD8+T cells and the depleting aged CD8+T cells in the mice. So what they did is they did the single nucleus RNA-seq analysis of these two conditions, and we have discussed some of the results in the previous video. And here we will understand that on the panel L and M, it shows that the differentially expressed genes mainly belongs to the single cell categories, that is either excitatory neuron or oligos, or even when we checked it within the excitatory neuron, we can find it belongs to either CA1 DG, mainly belongs to the CA1 DG, or the other cases in which the common genes that are dysregulated is very low, whereas individual genes that are dysregulated is very high. That is what it shows in the panel N and M. Afterwards, they check the differential expressed genes in the excitatory neuron, saying this is the one that shows maximum DEGs, and they are able to find out that genes that are dysregulated belongs to the categories of either metabolic pathways or synaptic pathways. And this thing they have found out using the gene ontology analysis, and we can see that they belongs to either synaptic pathways in this dysregulation, or belongs to the metabolic pathways such as this ubiquitinations, or this protein targeting to mitochondrial regulation of proteins. And so these are the changes that they are able to find out in the gene ontology analysis of this differentially expressed genes in the excitatory neuron. After finding out these changes, they want to validate in the form of actual responses. So they did the novel object recognitions, and also RAWM test of this mice model in these two conditions. That is, in one case they use anti-CD8 antibodies to depleting the CD8+ cells, and remember when they use anti-CD8 plus antibodies to depleting the CD8+ T cells, it is depleted only in the periphery or in the spleen, but not in the tumors. So when they checked it, when they checked the NOR time interactions, they are able to see that for the novel object recognitions, these anti-CD8 antibodies shows high time compared to the control antibody treatment, indicating that is with depleting of the CD8+ T cells, the mice become more familiar with the known object compared to the novel or new object. And similarly, when they check the RAWM test, they are able to see that this anti-CD8 antibodies shows less error for this RAWM test compared to the controlled antibody, it is IgG2b antibodies. So this further complements the results that they have seen in the panel M. That is, once the mice is familiar with the pattern or with the training, they are able to show less error compared to the case where anti-CD8 CD8 antibodies is not depleted, indicating this it's the aged CD8 antibodies that plays a role towards this. It plays a role towards this memory recognition or the cognition effect that is related to the brain. And by depleting these aged CD8+ T cells, they are able to recover or rescue some of these functions. This is evident with the high novel object recognition times. This also indicates that the mice that are familiar with this, more familiar with the old object. And so this is what they finally concluded from this panel figure 5, and this is the conclusions of this figure 5, that how they come up to the point where they show that the aged CD8+ T cells has a role towards this cognition effect. By depleting this aged CD8+ T cells, even though this depletion occurs only in the periphery, they are able to rescue some of the cognition functions. This indicates that the young or aged CD8+ T cells has a role. It's a completely different role So this is what we finally come to the point from from the end of this figure 5. If you need to understand more about the experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.